Distinguished colleagues, ladies and gentlemen, my name is Andrei Reshetnickov. I'm Ph.D. in Bioorganic Chemistry, and I'm representing the Academy of Holistic Medicine from Berlin, Germany. It's a worldwide organization offering holistic treatments for cancer in a variety of different modern methods, from orthomolecular to resonance and all kinds of other techniques connected with training the immune system and tuning the body to fighting with cancer. I'm going to present some facts about new product. It's called Singletin. This product is among the products and methods offered by the Academy of Holistic Medicine, and it's based on photosensitizer, chlorophyll derivative, Chlorin E6 derivative, and is used in course of the so-called photoimmune therapy. If we talk about immune modulation, then we need to say a few words how we come to this treatment and what is our background. The background for immune therapy of cancer, as well as photodynamic therapy, is the basis for it is surgery, because historically surgery was the method, the technique preceding. Historically, it was the first, and initially it was applied in the beginning of the 20th century, mainly to tumors that were on the skin. So the skin tumors were surgically operated quite successfully, and with time, with years, some medical statistical information was accumulated, so that by now we can say that with surgery about 10% of skin tumors on neck, body and limbs gives a partial response, as you can see on the diagram. Primary lesions of head cancers give up to 40% partial responses, and about 17% of the partial responses give recurrences. So this classification of surgical dissection of basal cell carcinomas according to Moss. Just in case we go for other methods of treating tumors, among them we can mention cryodestruction. It's using liquid nitrogen to destroy lesions, tumor lesions, by freezing. Up to 10% partial responses, like in case of surgery on the body, and up to 20% recurrences. So it's quite comparable to surgery, unless it's used on the head and neck. So with the head and neck, it gives a slightly better result. Then we can also mention electrical dissection. It doesn't have any use in case of recurrences, but for primary lesions, it can give us a quite serious response, up to 26%. So it's nearly the same response like surgery provides for the head and neck cancers. Since 1990s, laser therapy of cancer starts to develop, and laser techniques are mainly connected with infrared lasers, the so-called LITT approach, light-induced thermal therapy or laser-induced thermal therapy. It gives up to 4% partial responses. And in case of recurrences, it's just the second treatments, treating recurrences up to 15% partial responses. So it was a challenge to us if we could come to a solution to find some method or combination of methods, just if it's possible to come to a 0% partial responses, so 100% complete responses in one procedure. And if we may ask such a question, if there are no recurrences at all. So if just one single treatment is possible to reach complete response, so if just one therapy can result in no recurrences and in complete response. So it was our challenge, it was our goal, for which we went in as early as 1995, and we started with the photodynamic therapy as the new cancer treatment modality, following surgery, cryodestruction, electrotherapy, and laser thermal therapy. So, speaking about photodynamic therapy, we come from that field of bioorganic chemistry that deals with chlorophyll derivatives, the so-called chlorines. And it was our choice just because of a number of positive features about it, advantages of chlorines as compared to, for example, porphyrins or synthetic chlorines. So the choice of the natural chlorophyll derivatives was just, first of all, because it's cheap. Chlorophyll is the most abundant molecule on the Earth, and the actual availability of chlorophyll in the nature is megatons a year, and it's a renewable source. So every year you can get from the green stuff, green matter, any amount of chlorophyll you need as a raw material. So one can get chlorophyll from spirulina, just because it's just one type of chlorophyll available from spirulina. Otherwise, we can get it from nettle, urtica, or from any other higher plant. It's a very efficient way. So 1 kilogram of dry biomass can give you about 3 grams of active pharmaceutical ingredients, resulting in a product called Singletin. So you will hear this name, Singletin, through the presentation, as long as the Academy of Holistic Medicine is offering this product. So this product is based on chlorophyll, it's based on spirulina or nettle, but Singletin is not Chlorin E6 and is not chlorophyll. So when we talk about Singletin, we don't talk about chlorophyll or Chlorin E6. We speak about derivatives from chlorophyll and Chlorin E6. So there is a kind of a chemical process, a chemical technology to lead from chlorophyll or from the green biomass to Singletin. So from 1 kilogram of that, we can get about 3 grams of Singletin active pharmaceutical ingredients that is equal to about 100 intravenous doses of Singletin. Another advantage of the technology is that it is feasible. And there is a certain patent behind it. So it's a patented technology of one-pot synthesis of Singletin, active pharmaceutical ingredient, from pure Chlorin E6. In fact, the chlorophyll, pheophytine, and Chlorin E6 are used as the precursors for getting Singletin on a patented basis. The process of production is controllable. There are many control points and a GMP production facility in the Netherlands. So the finished formulations come from GMP facilities from both Netherlands and Germany. And the whole production process was developed initially and scaled up initially by us in the beginning of the 2000s. So as you can see, the renewable raw material production in case of spirulina or just nettle that can be collected from the wild nature. And the composition, the patented composition of Singletin is certain contents of Chlorin E6, Chlorin P6, and Pepperin 5. Those are 3 ingredients in the intravenous Singletin. As long as we are talking about photodynamic therapy, then we always need a light source to activate Singletin in the body. We use French-made laser Pharaon for 664 nanometers, so it's red light. It's a programmable laser device for photodynamic therapy and all adjacent therapies such as immune PDT or PIT or systemic light treatment. It's approved in the European Union for the medical use. So what about preclinical results? Just in a few words. Spectroscopically, here is the excitation spectrum of Singletin. It has 2 main peaks or bands. One is in violet area of the spectrum, the highest one, so it can be activated by violet light. And the other is at the red part of the spectrum. It's 662. And if you excite the molecule in any of the peaks, violet, green, orange, red, you will get fluorescence at 668 nanometers, so it can be used for the fluorescence detection of tumors. So, for example, this is a lesion of basal cell carcinoma of the skin. After intravenous administration of Singletin at about 0.5 milligrams per kilogram of body weight, so it's approximately 35 milligrams of the active pharmaceutical ingredient per an average person of about 70 kilograms of body weight. In about 2 or 3 hours, such kind of fluorescence can be seen using a fluorescence guidance equipment. So one can exactly see the border of the tumor, and for a surgeon it's easily to excise the complete of the tumor without leaving any residual tumor. And for photodynamic therapy, it's of course possible to mark exactly the circle around the tumor, to cover it with the spot from the laser for a complete eradication. So this is how we solve the problem of the complete response. If the response is incomplete, we can just repeat the procedure to the therapy to remove the residual cancer. However, in most of the cases, as far as we can see the border of the tumor, so we can completely destroy it using the laser and Singletin. So the advantage is that this peak, absorption peak, is rather high. It's much higher than for many other sensitizers, so that it allows for a very efficient activation of the molecule with light. High fluorescence yields, about 4%, means we can do not only treatment but also diagnostics of lesions, their imaging. High production of reactive oxygen species, about 70%. It means a high conversion of laser light into the reactive oxygen species, such as singlet oxygen, peroxides, etc., which play a crucial role in damaging cancer cell. And fast tumor accumulation in about after 30 minutes up to 9 hours, with the average time of 3 to 7 hours. The selective tumor accumulation takes place with the therapeutic ratio from 4 to 6. Fast clearance from the body. In about 2 hours, there is no Singletin in normal skin and muscles, so they are not sensitive to daylight, and skin around the tumor is not damaged by the laser illumination, and complete clearance from the organism in about 2 days, with 98% in 1 day and about 98% in 2 days. So there is no skin and eye and healthy tissue damage in the course of photodynamic therapy or after it, and there is no strong pain during photodynamic therapy. So in most of the cases, using of kind of shielding normal skin from laser light is not necessary. Neither it necessary to use painkillers in most of the cases. If we look at the pharmacokinetics and biodistribution of the active pharmaceutical ingredient by bulb C mice having embryo carcinoma T36, then one can see that after the intravenous administration, it makes a plateau between half an hour and 5 hours. There's the solid line. It's the accumulation in the tumor. While the contents or concentration in the normal tissues either stays low, so below the accumulation in tumor, or quickly dropping, let's say by 5 hours, dropping to nearly zero. So again, below the tumor accumulation. That provides for a high contrast index, therapeutic index, making it possible to treat the tumor between 3 and 9 hours, so in this range. Singletin concentration in organs and tissues can be easily seen here. So it's cleared from the body through the liver with the urine and feces, and it has high accumulation in urine in 30 minutes, so it accounts for a fast clearance. And in tumor, the highest accumulation, the highest ratio, tumor to skin ratio is 14, tumor to muscle ratio is about 3. So in fact, it's a huge difference between normal skin and muscle and the tumor already at 3 hours post administration. By 5 hours, the ratio in skin is 13, so tumor to skin ratio, so 13 times more in tumor than in the skin, and it's not detected in the muscle. So if we choose between, let's say, 3 and 5 hour intervals between administration of Singletin and photodynamic therapy, so giving light to a tumor, so for the skin, 3 hours is fine. For the mucosa, 5 hours is preferable. So for treating tumors of the stomach, esophagus, intestines, bladder, we should give light about 5 hours post administrating, so post injecting the photosensitizer. For the skin, it's possible to give light in 3 hours. And if we go for photoimmune therapy or immune photodynamic therapy, when we activate photosensitizer in the bloodstream using intravenous laser illumination of the blood, or using activating modalities when we activate the photosensitizer through its oxidation outside the body in the dripper, then we go for this 30-minute interval between infusion and activating the photosensitizer. So, in brief, it's just an overview about phase 2 clinical trials. It's been made in 3 hospitals with intravenous formulation, and in 1 hospital with superficial application of a gel. And I would say even phase 2 clinical trials took us longer than phase 2 clinical trials of the most of the anti-COVID vaccines. So it took us a few years here, even for phase 2. So each of the 3 hospitals who took part in the trial enrolled by 28 patients, and 30 more patients were enrolled for the for studying the topical formulation. What was the result of the clinical study? A small half a dose of formulation used for cancer, fluorescent diagnosis of cancer, and averagely high light dose of 300 joules per square centimeter gave us, let's say, it was called a light regime. So low concentration of photosensitizer and high concentration, high dose of light. This is the so-called light regime. That's why you can see a triangle, 1 and light. Then it's just a square with number 2, and drug means that it's the so-called drug regime or drug mode, when it's 200 joules per square centimeter light. So we don't pay much attention to the light dose here, but we give a high dose of the photosensitizer, 1.2. So it's twice more. And here we would like to see the influence. What influences the efficacy the most, the agent or the light? So to find what influences the most, actually we've chosen those 2 protocols. One with the maximum light dose and minimum drug dose, the other with maximum drug dose and minimum light dose. And of course, I would like to test actually influence of both high drug dose and high light dose. It's around with 3. So let's have a look at the results. and the results are quite amazing. So each of the hospitals, by 28 patients, used 3 regimes, and then they were combined in a blind study independent of the hospitals. So that was made by our company to randomize the data. It's just a kind of a description of the protocol of phase 2 clinical trials. So an optimal regime, optimal regimes with the efficiency data are presented here. As you remember, our goal was to reach 100% complete response, so 0% partial responses, after just 1 procedure. And quite amazingly, that we were able to reach that goal with the highest dose of the photosensitizer and the highest light dose, with observation at 2 months, 6 months, 12, and 18 months by biopsy. So it was just a follow-up of those treatments. After just 1 procedure, we have reached our goal. With other 2 regimes, there were partial responses. Here you can see about 20% partial responses with the so-called light regime, with a high light dose and low drug dose. It's quite, as you can see, it's quite comparable to surgery. And the so-called drug mode, when it was high concentration of the drug and low concentration of light, it was about 14% partial responses, so it was better. It leads us to a conclusion that drug concentration contributes more to the effect than the light dose. In fact, the more photosensitizer in the tissue, the better is the result. This was the conclusion after comparing those 2 modes. And in fact, if you see the structure, classification of the patients by morphological verification, there were more patients with stage 1 to 4 primary tumors without metastasis. Less patients with primary multiple cancers, with number of cancer lesions from 2 to 8, and still some patients with recurrences. So those were the patients enrolled in the study. So, in fact, what we could conclude for the intravenous preparation was that we were able to reach our goal, and then we switched to the topical formulation. It's a gel. The gel exposition was 3 hours. There was quite high light dose, higher than when we used the intravenous solution. And we treated, using the gel, we treated patients 4 times at 1 week intervals. So with the gel, the results were more modest, as you can see. In the light regime, there was no complete responses at all. In the drug regime, there were mainly partial responses. And when we combined again high drug dose and high light dose, there were mainly complete responses, so 0 partial responses. So in fact, again, with the topical formulation, we have proven that a combination of a high drug dose and high light dose is preferable with more influence of the concentration of the photosensitizer. So I'm now coming over to the conclusions. Following phase 2 clinical trials and the approval of Singletin in two countries, Russia and South Korea. We are going over to its bridging clinical experiments in the European Union, and aiming at the registration for treating skin cancers, superficial cancers, and also continuing clinical research in ophthalmology, otorhinolaryngology, gynecology, treating pulmonary diseases, including rehabilitation of patients having the COVID-19. So it's rehabilitation after the COVID pneumonias by inhalation of the solution. And we are going on with the approvals and patenting Singletin worldwide. So there are two registration certificates in Russia, South Korea. It was also approved in India as Ayurvedic product. Now I would like to say a few words about the current protocol to treat patients having stage 4, stage 3 progressing cancer, especially for palliation and for patients who refuse from the traditional techniques, or from patients who cannot be treated by conventional methods like chemotherapy, surgery, radiation therapy. So we can offer such an approach that the light, red light, is laser light is given not above the tumor, but it's given inside a vein. It's called immune PDT, or at the dripper while the photosensitizer is being infused. It's called photoimmune therapy. Both methods involve oxidizing the photosensitizer in the dripper or in the vein, and the oxidized photosensitizer, it has a potential of oxidation, so turning into an organic stable peroxide, thus modulating a specific immune response of a patient. So if you infuse a photosensitizer a few times and then illuminate the dripper or illuminate the vein simultaneously with administration, then an oxidized formulation accumulates in the tumor, thus oxidizing the cancer cells and changing the tumor markers. So it's a tumor recognition moment. So here you can see the whole protocol, but this treatment takes time because immune modulation is a slow process, same as tumor growth process. The patients are recommended to stick to a diet during this treatment, and minimum protein, low fat, so reduction in antioxidants while on this treatment. And in fact, I would refer to chapters 6 and 7 of my book, The Medicine of Light, where one can read about a diet and about those immune methods in more detail. So in about 2 to 4 weeks following treatment, tumor recognition takes place with inflammation in the tumor and breakdown process. And this is, we can also potentiate this response using other anti-cancer techniques like hypothermia, like treatment using herbs. And during this month, many toxic products may accumulate in the body, so we need a detoxification. This may be done using all routine ways of detoxifying the body, and we also can offer a diet. We can offer drinking more water and using drippers, using even chelation therapies and even hemodialysis. Sometimes some herbs. There are some products, herbal products, which we offer during this period of treatment. You can also read about it in our book, The Medicine of Light. And there are some food supplements we also recommend during those months. Month 1, echinacea, curcuma, soybean protein, selenium. During month 2, there are some products of ours. And during month 3, detox products, including enzymes and some products of ours as well, like Radoclean, Olga Romanov tincture, and so on. So thank you very much for your attention, and stay with us.